Thank you so much. This is our last panel of the day, and after that, we'll be all going to the pub, I think. Last panel, I think it's also a very, very exciting panel for myself. ATMP, Advanced Medicinal Therapy, Cell and Gene, MRNA. Right? I have 3 panelists here with me today. And first one, Dr. Lucas Chen. We know Dr. Lucas is always here on this panel, really, our usual guest. So Dr. Lucas is the Chief Scientific Officer of CellVec in Singapore. They're a CDMO dedicated to the development and manufacture of the viral vectors for human gene therapy. Lucas received his PhD at Imperial College London and was a senior investigator and head of manufacture for advanced therapies at King Health Partners in London, where he held the translational development of ATMPs for the clinic and established the UK's first GMP viral vector core. He is an elected fellow of the Royal Society of Biology and advisory member of Singapore's Ministry of Health on cell and gene therapy regulations. Welcome, Lucas. Second panelist, we have Kwangjun Yoon from Korea. Welcome to Singapore. KJ is the managing director of the BioGMP CHA Biotech from Korea. He is responsible for cell and gene therapy CDMO, and he's highly motivated, creative, and global-oriented process manager and project manager. He has over 20 years experience in the pharmaceutical industry from Janssen Vaccines, Samsung Biologics, SK Chemical, and CHA Biotech in Korea. He's very effective working based on the knowledge of 3 areas of process, development, manufacturing, and engineering. Last panelist, we have Peijing Zhang with us, and thank you so much for accepting to be on this panel last minute. It's
my great pleasure, definitely.
Thanks, Peijing. We worked with Peijing in the past, and he is currently the strategic technology partnership leader for gene therapy at PAL Corporation. And he is responsible for technical engagement with customers and gene therapy community across Asia Pacific region. Peijing has more than 15 years of experience in biotech research, development, and commercial manufacturing. Prior to joining PAL, he held various roles with increasing responsibilities in tech transfer, manufacturing science and technologies, and novartis, and analytical science and CMC management at Transcenta. Before moving to the biopharmaceutical industry, he was with A-Star, Singapore's Bioprocessing Technology Institute, and was an assistant professor. You were there? At the national
adjunct assistant professor, to be stringent.
I wanted to skip that word. Peijing obtained his PhD from the NUS, and he has published more than 20 peer-reviewed papers in biology and biotechnology, and is a frequent speaker in international conferences, including the ISPE conference. Thank you so much. Okay, let's start this panel discussion. So, I think, firstly, I want to ask, Peijing, I know you from PAL, but Lucas and KJ, you all have your own product, and I believe there are a lot of things in your pipeline as well. Maybe you can give an introduction. What's so exciting in your pipeline?
Okay, I'll go first. So, a very brief summary and introduction of our company, CellVec. So, we're mainly focused on cell and gene therapy space, particularly in viral vector development and manufacture. So we noticed quite early on a significant bottleneck in access for clinical grade and GMP grade viral vectors for clinical deployment. So CellVec is a CDMO, and we provide end-to-end solutions for viral vector from molecular design all the way to GMP manufacture. So our solutions is integrated. We have expression constructs, to a fully qualified master cell bank asset, all the way to an integrated up and downstream manufacturing process platform, which are wholly developed in-house. So we have a newly constructed and qualified and now GMP certified manufacturing facility based in Singapore, in Pasir Panjang. We're very proud that to receive this certification and accreditation, being the first in the region, which we have been accredited to manufacture both retro and lentiviral vectors for clinical supply. So what we're hoping to alleviate is the bottleneck to accelerate clinical development, particularly in cell and gene therapy. And I think, as we come out of the pandemic, we are hopefully getting some relax away from the focus on the pandemic therapeutics or vaccine development, moving back towards a focus and energy to reignite, this backlog of therapeutic development. So that's one of the key areas of CellVec's interest, and other areas surrounding key gene transfer technologies.
Thank you. KJ, please.
Okay, my gratitude to introduce our company, CHA, Cha Biotech. Cha Biotech is a very unique business model because Cha Biotech started a hospital, and then they established a university, and then also to establish a research institute, and then finally we established companies. So Cha Biotech have 20 years experience developing cell therapy, specialized stem cell. At this moment, we have 3 products in the clinical study stages. The 2 is stem cell based on the umbilical cord derived MSC, mesenchymal cell stem cell, and another is the immune cell, the NK based cell therapy. And Cha Biotech established the cell library technology platform with the umbilical cord and also placental to stem cell and embryonic stem cell, and also the adipose-derived stem cell, and also NK-based. With this technology platform, we expanded our business to CDMO business as well. For the cell line development and also the clinical material production and also commercial material production, is successful launching, going well. Thank you very much.
Thank you so much. We see a lot of requests recently, all this like hospital, and they transformed into commercial manufacturing of viral vector or cell and gene therapies. So talking about the trend, also, Lucas, you mentioned post-pandemic, what you think is the current trend and status of this industry?
So I think what I'm hoping and to see is that the significant resources strain that I'm sure many of us manufacturer to developers have experienced on the pandemic to the whole ecosystem, from supply, raw material supply, to drug development being halted, to clinical trials being halted, to cancer drugs development being halted, to see perhaps a flatlining of the pandemic being so acute and draining resource, not in a negative way, but obviously in a necessary way, to have a bit more time and resources and to rededicate the energy back to therapeutic development. So that's what we are beginning to see, and hope that this pathway will continue. And as we learn from the lessons on how CDMOs, manufacturing, drug development deal with the pandemic, and how we learn lessons on resilience, agility, perhaps in the next pandemic or the next emergency use, and rededicate our resources, that perhaps we could be more balanced in the way we deal with resources, in the way we deal with drug developments and situations like that.
Sure. KJ, would you like to add some?
I hold FDA the IND submission the past 3 years during corona pandemic situation. So every industry is depressed almost, but for example, cell therapy, the IND submission and also gene therapy IND submission is increased dramatically, almost 3 times during that time. That means maybe cell and gene therapy will be maybe third wave of bio-revolution appearance. So, and at this moment, T-cell based therapy will be increased for the process development. So, maybe some the viral vector or the the vector system to be necessary to more and more. So actually, but the problem is we have to prepare a large scale demanding on the market. So that is that's why we need some the application innovation to increase the capability. That is the current issues.
Yeah.
In my opinion.
Yeah, that's great. So technology, Pei-Xing, that's your area. You have anything for the trend and status?
Yeah, sure. So maybe before that, just to add on to what Lucas and also KJ shared in terms of the market dynamics over the current period. I think I agree with what Lucas and also KJ shared. Indeed, the pandemic actually has caused a lot of disruptions in terms of supply chain and also the focus, right? The drug development focus. So indeed, I actually provide a lot of technical consultations to the customers in the APAC area. So definitely we can sense, there's actually a divergence of the focus away from viral vectors, for example, for, genetic disorders and things like that, to shift the focus more on the pressing need on combating COVID-19, right? So indeed, we do see a fair share of interest in mRNA technologies and also related viral vaccine technologies, right? But I think that's actually, causing some disruptions or kind of a shift of focus. However, I'll also say, I think that the other two panelists would agree, the pandemic also catalyzes a lot of new thinking, on the drug developer side, technology side, and also fostering some strategic partnership with solution providers like ourselves, right? So I think earlier on in the workshop, I was actually sharing with the audience our partnership with Oxford University, AstraZeneca, to come out with COVID vaccine based on chimpanzee adenovirus. So I think that's actually one of the examples of how the disease actually, positively catalyzes the change, a paradigm shift in the industry. On top of that, I would like to share there are additional shaping forces or driving forces to, the situation that we are right now. I think in the APAC area, I think it's fair to say that we are having, a relatively slow start in terms of viral gene therapy, mRNA side of technologies as compared to Europe and US. I think Lucas can comment on that because of his, experience back in the UK. So a lot of investment is actually being quite cautious to wait for the demonstration of proof of concept for example, viral gene therapy, right? And I think a lot of the people in the audience would actually recall, especially last year and earlier this year, there are a few kind of a safety warning or potential warning signals that put a lot of drug developers on the wait and see kind of a mode, right? So I think that's another driving force in terms of the current market situation. But I think most of you have read the news yesterday. It's actually very exciting, the new approval of gene therapy from Bluebird Bio. So I hope that's actually kind of a, is putting a, good momentum to shift, as Lucas said, the focus back to developing drugs to solve the unmet medical needs.
Yeah, absolutely, and absolutely good news. But from this good news, I want to come back to Lucas's point, and you mentioned disruption of the supply, etc., etc. What about the challenge of starting material? And you are providing one of the starting material to the cell therapy, gene therapy area, and KJ is kind of like on the receiving end. And how you two see it differently?
Well, I think I'll just following the point that you know, the pandemic has done bad things, but you know, it really taught us a lot of great things as well. I mean, one of the key lessons I've learned is, you know, it forces us to rethink everything from technology to development, technology development to working together, to collaboration, to accelerating, you know, data review, speaking to regulators and being more transparent. It really pushes us to step out of our traditional comfort zone and risk-averse situation, to speak to regulators, to become more transparent with the way we share data with the general public as well. And, you know, it comes back to, you know, the risks of testing new medicine in drug development, the risk involved. And, you know, I'm hoping that the traditional therapeutic development can ride on that new understanding, you know, with regulators, for example, or the general public in testing new medicine, and not be so afraid, and maybe developers and manufacturers to be more open in describing risks, describing challenges that we face. So that's what I'm hoping, that we could take that lesson on. From our perspective, in terms of viral vector manufacture, the issues that we faced before the pandemic is now coming back to the forefront. You know, the Bluebird drug is very encouraging. You know, it is very expensive treatment. So these same problems is now coming back again. Cell and gene therapy is extortionately expensive. So we need to find ways to simplify it. We need to find ways to reduce the cost of goods, particularly looking at the demographics of Southeast Asia, where, you know, affordability is obviously a main issue. But, you know, the silver lining is that cell and gene therapy development is going through the same cycle as traditional biologics, small molecules development, where initial cost is high, but hopefully over time we find efficiency in development. For bluebirds, for example, it sounds really expensive, but you know, it's a one-time cure for the removal of a need for transfusion. So it's a lifelong cure. So, so there's that balance in looking at it. But yes, hoping that we can go back to drawing board to tackle the needs on cell and gene therapy and realize its therapeutic potential.
Sure,
Yeah.
KJ.
Yes, the cell therapy matters. Starting material is the cell itself or the blood from the hospital. So actually, starting material is required to oversee the product lifetime. So actually, the tissue or the blood collected from the patient at hospital or the clinical center. So actually, the traceability control is very important. So the GMP controls should be started from the very early stage in the hospital, from the clinic center. There's also common the contamination risk with bacteria, mycoplasma, and water fungi, and also the adventurous agent, including the virus, something like that. This kind of the contamination risk should be mitigated before receiving at the GMP facility. But in generally, the hospital don't have generally the GMP facility inside. So sometimes it's impossible to perform the aseptic condition, the manipulation the hospital. This is a very big challenge. And also, the donor eligibility procedure should be in place from the hospital or the clinic center. So now, so actually, medical assessment or the donor health check history should be secured under traceability control. That is kind of GMP procedure. Yep.
I think two things you mentioned. Your product is autologous, right?
Autologous
Yeah.
stem cell based.
Yeah. So the nature of your product and the nature of your company is coming from a hospital kind of a setup. So the scale of your current manufacturing, it will be a question mark. How you plan to scale up or scale out of your manufacturing?
So for autologous the cell therapy, it's very challenging to ensure the scalability because actually there's two types of the cultivation method depending on cell type. So first one is the suspended cell. The suspended cell is more the close to conventional the scale of the approach because using the bioreactor system, maybe we can make some the scale up the approach for ensuring the scalability. But problem is adherence cell type because adherence cell type is very hard to implement to some scale up the matter because we collected the tissue from the hospital and moved the GMP facility. And then we have to cell isolation and also enzyme treatment and cell isolation. And then we have to perform the primary cell culture and then going to main cell expansion, the processes. So actually, fully the integrated processing is a little bit impossible at this moment. But recently, I saw some green light from some provider because they presented some the practical approach how to make some scalability for the adherent cell therapy. So at this moment, current technology will not possible to achieve all automated or the integrated solution for adherent cell cultivation. But new technology will come and with some good best solution for it. Okay.
Yeah. Just extending on your last note, and the new technologies, once the technology is, and there will be automation, AI, etc., maybe the other two panelists can comment on that as well, starting from Pei-Qing.
Yeah, so maybe to comment on new technology adoptions for new modalities. So I think, first of all, I would say if you look at the current technologies for making viral vectors, and also mRNAs, right? From a processing point of view, actually there's a lot of readoption of existing technologies that were designed, manufactured, built for mAb processes, right? The conventional bioprocessing. Are they the right tool? Are they optimized with the optimal process parameters? Are they at the right scale, right? For example, very specifically recovery, right? Non-recoverable volume and things like that. So I think there's actually a lot of room to actually invent and innovate, improve, to make sure that we actually come out with, again, connecting to this Malding's topic in terms of modular design and things like that. Make sure each of the module is actually optimized as much as possible, right? The right tool for the right job. So I think that is that. And secondly, of course, as you were actually alluding to, how to connect those systems efficiently with digitalization, automation, right? I think that's definitely big areas for improvement. Yeah.
Thank you.
But I think looking beyond processing technologies, I think the audience would agree. I'm actually talking about cell culture-based modalities like viral vectors. So I think if you recall, in the early 2000s, when I was still a graduate student back then, there were still groups or companies doing transient transfection of CHO cells or HEK cells to produce the amount of antibody, gram level, right, to enable you to perform like pre-GLP tox studies or candidate selection. Now it's actually getting much rare, right? You can quickly come up with a stable pool and then stable clone to enable your GLP tox and CMC program. So I think on the viral vector side of things, the key is also on the biology side. how to optimize the expression vector, and how to, I think there's also a lot of speculation on getting stable cell lines for virus production, or at least stable packaging cell lines for virus production. But do we already have kind of a gold standard proven tool in the market? I would really doubt so, right? But definitely there's a research area that we should focus on in addition to processing technologies. So in short, a lot of research is needed on the biology side of things for us to really, have a quantum leap in the innovation. That's what.
Yeah, that's great. I think Lucas, you can't wait to.
No, this is really valid points, and I follow on from Peijing's points about repurposing. I mean, particularly in the viral vectors, which in a way is we have the luxury of being a lot more simple compared to autologous personalized therapy in terms of scaling. But even that itself is extremely complex, and we've been borrowing technologies from MAPS, from monoclonal antibodies, and I'm just beginning to see, and I've been pleading with technology providers to come up with more customized solutions. But I'm beginning to see really nice new technologies, from scaffold design to bioreactors on upstream side for viral vectors to downstream. I've seen some very recent development on viral vector specific type of affinity chromatography, which is really nice to see, and also the point about stable cell line development. Ultimately, the drivers is about scalability, about standardization, is about building robustness in the manufacturing process. And I think for cell and gene therapy is a particularly challenging area, because a lot of these early clinical development actually came from an academic lab, from a university, whose perhaps priorities and objectives and concepts is not even about speed to market, but perhaps speed to the first IND filing. So perhaps a lot of times not enough attention has been paid towards manufacturing process development, analytical technology development. So those are all the challenges that makes this field more exciting to be in. Yeah, but those are critical areas that we'd like to address.
Definitely. Maybe eventually one day we'll see all these robots manipulating the cells. Despite all this really exciting, great technologies out there, our industry is still very highly regulated. So we still need to come back and talking about the compliance regulation. What you think the current trend of all this regulation development? Maybe Lucas, you can start.
I think in terms of regulations, we've seen FDA issuing updates on chemistry manufacturing control guidance on viral vectors twice in the last few years, and we've seen FDA talked about issuing new guidance on cell and gene therapy as a whole. And I think that's a very, very much welcome, because there's still a lot to learn from cell and gene therapy, from how the therapy behaves, from identifying the key quality attributes of what constitutes the therapeutic modalities. So Singapore ourselves have now a new cell and gene therapy regulations, which is much needed, and it's really nice. It particularly addresses points on early phase development, so we can help early developers to translate and transition into GMP manufacture and clinical deployment. So I'm very positive to see this route continuing to expand.
Sure, KJ, you see the same in Korea?
Yes, just interesting is Korea regulation, because actually there's two types of pathway for the cell and gene product for patient to use. One is regenerative medicine. So another is biological product pathway. This is different, so absolutely. But in Korea, 2 years ago, we have government announced just one, the regulation, the law, on the law, the combining for both, advanced, the regenerative medicine and advanced biological product. But when I saw this regulation first, a lot of conflicting things because unclear the boundary, scope of the law, and also there's some not enough information how to practically approach the pathway, through the pathway for the development. But so government made another interface, the discussion, the communication, the tool. We call it the dynamic bio. So they hold every the meeting and invite everyone, and then everyone can raise the issue, and then the government to have us mind all the issues and published their statement in a very specific way. This is very good communication harmonization tool in Korea. Yes.
Any impact on you, Pei-Ching?
Yeah, definitely. So I will share maybe two thoughts. One is actually the challenges the industry, cell and gene therapy is actually facing in terms of regulatory affairs, right, or compliance side of things. And secondly, I'll share what we can do, especially us, Paul and also our sister company, Cytiva, can do, as solution providers to join forces with MAH, with CDMOs, right? So the first part, so regulatory challenge, I think it's kind of a paradox. If you look at cell and gene therapy, a lot of them are actually either received or aiming for accelerated approval or similar designations. For example, the RMAT in the US FDA, the PRIME for EU or EMA, and also Sakigake in PMDA Japan. And similarly in Korea, there's also accelerated regulatory pathway, I believe, right? So that is that, right? So there's an opportunity for you to bring your product earlier to the market for unmet medical needs. We're talking about genetic disorders, orphan drugs, and things like that, right? So those are potentially life-saving. But the challenge is really, if you look at cell and gene therapy, we talk about huge room for improvement on the processing side of things, on biology. And this, when you put these two things together, now right? So from a program level, you are actually put on an accelerated regulatory path. But on the other hand, there's still a dramatic or perceived lack of data, compressed timeline for CMC development, right? So these two put together presents really tremendous challenges to drug developers and CDMO even included, right? Secondly, I'll share how we, as solution providers, can support, to help the industry to debottleneck. So we know there are various professional bodies, societies. So for example, in the US, there's ARM, right? And there's also NIMBLE. So, just by the way, Paul, Cytiva, we are actually active members of NIMBLE. And through NIMBLE, we can also weigh in to share our platform knowledge. We have actually worked across the industry, multiple customers, multiple projects, and based on our technology, what is the control points, what's the parameter ranges and things like that. Specifically, if you look at ARM, there's actually a project AGENE. So it's actually similar to AMAP, A vaccine, right? So which actually provides you a nice QBD framework, right? Which the MAH or CDMO could leverage upon to jumpstart. And similarly, at Paul, we published QBD for AAV, where we actually comb through the process steps, right? From bioreactor all the way to sterile filtration, where we actually highlight, based on our field knowledge, these are the CQA or impurity-related CQA. These are the CPPs, and these are high-level the ranges, right? And hopefully this will also help our users to avoid some of the pitfalls, to accelerate, and also to stay compliant to GMP regulations.
Yeah, I think that's a great emphasis on the collaboration in the industry.
Definitely.
Yeah. So before I turn the questions to the audience, I just want to have the last final question to KJ, because you have to finish the product, and because of the intrinsic nature of your product, you can't really wait until all the release test results available, but you have to inject your product in the patient. So how you actually harmonize this with the regulators and within the industry?
that is a very, very big challenges on autologous cell therapy area. So depending on the cell therapy, so different analytical test method is used to determine the product quality. But frankly speaking, the cell therapy area, most common, typically bioburden test and endotoxin and cell viability, and then so on. Facts, yeah, flow cytometry test is necessary for the final batch release test. But autologous the product have a very short lifetime. For example, maybe within 24 hours, maybe less than. So if we consider the perfect bioburden test, maybe as you know that the bioburden test normally takes maybe 14 days, something like that. So we need another the final test, the method. So currently, the industry announced maybe rapid bioburden the test method. But this rapid test is a limitation with some specific species detectable, and also very strictly the customized test method validation is required. So the many companies consider the steroid approach. For example, 2 steps of final release is maybe first of all for bioburden test, maybe rapid test is the first the final release, and then finally maybe 14 years, maybe that is the final release test. But the absolute for any cases, maybe national scientific, national, and also database justification is essential to ensure the quality system.
Thank you so much. Now feel very much assured with the quality. So I would like to invite our audience to have some questions to our panelists, or you have any comments and discussions you would like to have? Or any final comments?
Can I just add the point to KJ's comments about analytical testing and standardization? You know, equally for viral vectors, things are still very customized and non-standardized. So you know, we touched on about regulation or regulatory supports, and we're just beginning to see perhaps some harmonization on analytical technologies. And I think it's really nice to see solution providers to guide developers from an early stage to implement robust and standardizable or scalable solutions from early on to tackle quality with quality by design principles from an early stage. It really helps CDMO in terms of scaling up and commercialization. Yeah, so that bottleneck from an early stage is really important.
Sure, thank you. Pierre, yeah.
I'll try to elevate myself to the level of the panel. So we talk about the COVID, and you explain the regulatory framework. We'll talk about the patient that need your product. What is the current situation in the health policy to help develop those kind of product, and is Asia the best place to do it? And how do we get access to those medicines? I compare to, for example, the COVID vaccine, where the whole world agreed that there was going to be a substantial government support to develop the vaccine. So are you exposed to that? And what is the current situation?
It's more like the government policy, right, rather than the regulation. So if there's any policy like help the industry to accelerate or even initiate, if you think Asia has the best policy, we have the best environments to do so.
Maybe I throw my 3 cents and then to invite Lucas and KJ to share their expert
Do you
opinion.
need to add a disclaimer? It's only your opinion. It's not
my
power.
personal. I'm not a politician, by the way. I think in terms of where is the best place to do business, again, my personal opinion is we really need global collaboration, stronger than ever before. I think we are in an increasingly sensitive period where we are seeing those invisible, tensions, right? I think global collaboration is really the key to bring the product to the market and also to bring the products, accessible to different populations. So, for example, the Oxford AstraZeneca vaccine, as an example, right? It actually originated from University of Oxford and then pushed to regulatory approval by solution providers, CDMOs, pharma as AstraZeneca. But then it's actually got quickly tech transferred to developing countries as well. I think yesterday we were chatting about it. There's actually a number, right? This is actually, perhaps one of the most, in terms of the volume, the largest vaccine that's produced for COVID, one of them. So I definitely see, this is actually one of the benefits for global collaboration instead of doing things in the region, only for that region in the silo. That's one cent I throw. The other is really, I think I'm referring to the ISPE journal, the Pharmaceutical Engineering, published earlier this year. When you look at the data, there's an article on the acceptance rates of common control strategy. So the article analyzed the acceptance rates in Canada, in EU, in FDA, and Japan. It's actually, my opinion is disappointingly low, right? The common CMC control strategy is not accepted in most cases for biologics. And if you extrapolate it for new modalities, it's not looking very positive. However, the article actually ends saying that if you look at common control strategy for mRNA vaccine, every country is accepting it. So why is that we can do it for mRNA vaccine for COVID, but cannot do it to other life-saving medicine? So I think that's another area where global cooperation can really help address unmet medical needs.
Sure, thank you.
Yeah, it's I kind of elevate myself to be careful about the regulatory level on the regulatory approach from the region, and that's why I think we can take a lot of positiveness from the pandemic on pushing boundaries on regulators to evaluate unmet needs, emergency medicine use, to hoping that momentum will continue back into therapeutics in the way the general public and regulators look at risks, look at the unmet needs to particular indicators and conditions. So I think it'll be interesting to see maybe the next couple of years as we come slowly out of the pandemic, as it really a test period to see whether we fall back into our old ways of being risk averse, being safe in our seat, or do we take that momentum forward? And I'm hopeful. I'm hopeful that this international collaboration, including Southeast Asia for cell and gene therapy, which is behind, from Europe and US, to take that momentum forward.
Sure.
Reluctant to say my opinion because this is very sensitive matter. But as I mentioned before, I think I'm a little bit specialized stem cell. Stem cell development, research and development is very difficult because the limitation with some MOA characterization is unclear. And also source material is the limitation with some variability. And also manual operation is there, so a lot of manual operation. But as I mentioned before, Korea announced just one guideline for both the regenerative medicine and also the biological medicine. Looking for the regenerative medicine pathway is a little bit simple and accelerated. The track is to develop. So actually, it will be very good model, a fast track model for the all of the world. If Korea this rule is realized practically, yeah.
Right, thank you so much. With that, actually, I want to finish our panel with quoting Lucas's word, hopeful. Let's end this panel with this word, hopeful. This is the end of the conference for today, but we will have a drinking session starting from 5:30, right? I think so, 5:30 to 7:30 something. It's the bar across the road, Millennia Walk, Tap. Yes, yes, okay. So hope we see all of you there. Okay, thank you so much for coming.
Thank you.